First question, Griffith transformation experiment. In this way, Griffith worked on bacteria called Streptococcus pneumonia. Streptococcus pneumonia. In this, there are two types. One is S type, another one is R type. S type, it is smooth and shiny, whereas R type, it gives rough colony. Smooth is virulent, whereas R type, it is non-virulent. Rough strain, rough colony, unable to cause disease. Griffith worked on a mice in 4 different ways. At first, he did where he injected a strain into a mice, where mice suffered from a disease and it got died. In a second experiment where he injected a strain in a mice didn't get any disease, it remained healthy, so mice live. In a third experiment where he has used heat-killed S strain and injected in a mice, where the mice lived. In a fourth experiment, he did a different thing where he has used a heat-killed S strain and it combined with R strain. He injected in a mice, heat-killed S strain plus R strain, inject in a mice. After injecting, mice got died. Based on this experiment, he concluded that something is being transferred from heat-killed S strain and to the R strain, so that mice got a disease and it got died. Second question, what is genetic code and salient feature? So where we'll find amino acid sequences. So there are 5 different characters. First one is triplet nature. Genetic code has a universal nature, it has degeneracy nature, and also it acts as an initiator codon, and also it has a terminator codons. Triplet codon, where genetic code is always present in the form of triplet nature. For example, CCC, ACC and AUC. Universal, so the genetic code will be same for all organisms, both in prokaryotes as well as in eukaryote. It will code for a single amino acid. Degeneracy, more than one codon code for a single amino acid. Example, proline valine. Initiator codon AUG, that is methionine. Terminator codon, it terminates the translation UAA, UAG and UGA. They are the terminator codons. So here initiator codon AUG, it codes for methionine. Then terminator codon, there are 3 codons, UAA, UAG and UGA. So they will stop the process of translation. Next question, explain the double helical structure of DNA. So this DNA X-ray diffraction structure was given by Wilkins and Franklin, Rosalind Franklin. Then double helix model was given by Watson and Crick. Salient features of DNA, where the backbone of the DNA is mainly made up of sugar and phosphate, and the 2 chains are anti-parallel in nature. One is 5' to 3' direction, another one is 3' to 5'. Then 2 strands are held together by hydrogen bonds. Then adenine always pairs with thymine through 2 hydrogen bond. Guanine binds with cytosine by 3 hydrogen bonds. And 2 chains of the DNA, they are coiled in right-handed fashion. And also pitch of the helix is 3.4 nanometer, that is 34 angstrom. There are roughly 10 base pairs in each turn. And distance between the base pair inner helix, that is 0.34 nanometer. So this is about your double helix structure of DNA. Next one, draw a diagram of nucleosome and explain the packaging of DNA in eukaryotes. So this is a typical structure of a nucleosome. So nucleosome is mainly made up of two different components. One is histone octamer and a DNA. So histones, they are positively charged, whereas DNA, it is negatively charged. Histone octamers mainly contains two molecules of H2A, H2B, H3 and H4. So togetherly it will form a octamer. Histones, they are rich in basic amino acids like arginine and lysine. Histones are positively charged and they are rich in basic amino acids like lysine and arginines. These histones, they are organized in histone octamer, where it consists of two molecules of H2A, H2B, H3, H4, as I have already mentioned. DNA is negatively charged, whereas histone is positively charged. Together it will make nucleosome. The nucleosome contains 200 base pairs and nucleosome will form a chromatin material, and these nucleosome, they will be seen in an electron microscope as beads on a string. And these chromatin fiber, they will make a future chromosomes, and in a chromatin we will be seeing two different type of chromatins. One is euchromatin, other one is heterochromatin. One is lightly stained, other one is darkly stained. Next one, Hershey and Chase experiment to prove DNA is genetic material. So in order to prove this one, they have used a bacteriophage. Bacteriophage are the molecules which attack or it will infect a bacteria. When they infect bacteria, they will penetrate their genetic material in a bacterial cell. And during that, they will remain their viral coat outside, and genetic material will be injected and it will get integrated with the bacterial genome, and they will multiply within a host machinery, where they have used two different materials, where one is radioactive phosphorus, another one is radioactive sulfur. In this one, they grew bacteriophage in a media containing these two, radioactive phosphorus and radioactive sulfur. After doing that one, in order to observe whether the DNA contains a phosphorus or a sulfur, whether the DNA is the genetic material or protein is the genetic material, they have used a agitation and also blending method to prove the DNA is the genetic material, and it is very important for your examination.